Yeah, so thanks for having me. I believe that Chem Talks was a tradition that started after I left the department, so it's a great pleasure to actually be able to give a presentation here. And pardon if my brain is a bit slow. It's only 6 in the morning here right now, but I'll try to be as clear as possible. The work I'll be showing today is on 2,6-dioxyglucosylations, reactions that are catalyzed by these bis-thiourea catalysts. I'll give you more details further in the presentation. This whole project was in collaboration with a very, very talented PhD student called Peyton Beyer. We're both part of the Jacobson lab at Harvard. So let's get into it, and keeping in with traditions of the Chem Talks, let me just see. There we go. I have a slide here first on my background. So I started out doing my bachelor's in chemistry at the University of Copenhagen, and then later started a 4+4 integrated master's and PhD program with Christian Marcus Petersen as my supervisor, which ended up in this thesis called Carbodium towards the ideal glycosylation. And along the way, I had the pleasure of engaging in a number of different, very, very fruitful and interesting collaboration, namely with CAS Taiyuan, which is a university in Shaanxi, China, where we collaborated with professors Chao and Wang on some very interesting, more mechanistic studies. And then I also was part of a collaboration with Novo Nordisk, namely a researcher named Rolf Tonning on automation, which was really, really interesting. And some of my favorite memories from my time at the University of Copenhagen centers around both teaching and mentoring students, but also my involvement in the Chemie Review was something I really have fond memories of, and also a journal club I was part of called the Dead Chemist Society. And then during my PhD, I had the pleasure of spending 6 months in the Jacobson lab here in Boston, and returned as a DFF International postdoc, which I'm doing now. And I can also already tell you now that I just received a fellowship from the Carlsberg Foundation to do an independent postdoc as a Carlsberg Reintegration Fellow from April. So I'll be giving a little teaser of what I'll be doing when I return to KU this spring. Anyways, let's get into the actual science. So for those of you who know about the Jacobson lab or may have heard of it before, will know that we mostly focus our work on enantioselective catalysis. And if we go back to 2008, Sarah Wiesman and Abby Doyle, pretty impressive team to have working on a project, were working on optimizing this enantioselective addition to oxycarbenium ions. And back then, they looked at this starting substrate and figured, well, if we're doing additions to oxycarbenium ions, isn't that essentially doing glycosylation chemistry? And it's pretty cool if you go check the supporting information of this 2008 paper, you can actually find that they tried doing glycosylations with these catalysts. However, we realized that these catalysts are not very competent at stereoselective glycosylations, and a few years would actually go by before it was realized that, first of all, in many cases, these catalysts are actually not monomeric in the transition state, and therefore students in the lab started searching for dimeric versions that wouldn't just aggregate in solution, but where we actually designed the catalyst to have 2 hydrogen bond donating motifs rather than just one. So these are the bis-thiourea catalysts that I'll be talking about today. We commonly refer to them as Naomi dimers to honor Naomi Rajapaksa, who was the first to successfully synthesize one of these catalysts. And not long after, a macrocyclic version of a bis-thiourea catalyst looking like this was actually shown to be a really efficient catalyst in stereoselective glycosylations, using glycosyl chlorides as the electrophiles. They were able to achieve a lot of different, very, very impressive glycosylations. Shortly thereafter, it was realized by a team of very talented grad students in the lab that phosphates are in fact significantly better substrates in this type of transformation. So they realized that using phosphate leaving groups rather than chlorides actually results in significantly higher catalytic efficiency. And based off of this now phosphate-based glycosylation platform, and still relying on these dimeric hydrogen bond donor catalysts, we've been able to achieve highly selective beta-galactosylations, beta-mano and beta-rhamnosylations, beta-furanosylations, and more recently, this paper that I'm still completely mind blown by. Of course, I'm biased, but site-selective beta-glycosylations of minimally protected sugars. So in this case, they glycosylate a triol with selectivity for the 2 position. I encourage anyone who's interested in physical organic chemistry to go read this paper. It's really an impressive piece of work. So a model, I just want to take you through a very simplified model of how we believe these glycosylations work, and I believe we have good evidence for this. So these catalysts are bifunctional and have a hydrogen bond donating part, which serves to activate the leaving group, and has a hydrogen bond accepting part, which is an amide that serves as an activator of the nucleophile. And we believe it acts as a general base, at least that's what most of our evidence points towards, to facilitate these stereospecific glycosylations very much, which has a lot of similarities to how enzymes actually behave in glycosylations. And the real key to achieving these highly selective glycosylations is correct matching of nucleophile and leaving group activation. Because if you have too much of one or the other, the catalyst can simply just self-aggregate. I'm going to be talking about 2-deoxyglycosides today, and 2-deoxyglycosides are really important constituents of natural products. They show up in a lot of bioactive compounds, mostly bacterial natural products, as you can see here. I've just highlighted all the 2-deoxy beta glycosidic linkages in all of these compounds. So these are a class of really interesting compounds that unfortunately are very, very difficult to synthesize. And the central challenge in synthesizing these, if we just look at a simplified reaction scheme, I've just drawn here a generic 2,6-dideoxy sugar, so you can see it's deoxygenated on the 2 and the 6 position. If we just do a glycosylation of an alcohol in presence of some promoter, we get an alpha glycoside and a beta glycoside, which will typically be equatorial. And this is the compound we're interested in. The problem here is that it's really, really difficult to control this substitution reaction because these deoxy electrophiles are really, really reactive. And a good measure of what reactivity means was generated by the Wong lab back in the late 90s. They developed this that they call relative reactivity values. And this essentially is just measuring the rate of methanolysis of various different electrophiles that are commonly used in glycosylation chemistry. These reactions largely proceed via various SN1-like pathways, so either via intimate or solvent-separated ion pairs, highly reactive intermediates, or these other ion pairs you could imagine formed depending on the protective group scheme. And if we search through, so there's a lot of data on relative reactivity values for various electrophiles out there. If we look at what factors that influence given electrophiles' reactivity towards SN1 pathways, we see that configuration, that means going from, in this case, mano to gluco and then to galacto, we can see that the relative reactivity values that I've shown here only vary with about 1.5 orders of magnitude. So that's not the greatest change you can get. However, switching protective groups from ester protective groups to ether protective groups has a huge impact on the relative reactivity values, changing about 3 orders of magnitude. And in addition, deoxygenation has a really incredible impact on this. So deoxygenating the 6th position affords about 1.5 orders of magnitude, whereas deoxygenating the 2 position gives about 3 orders of magnitude in reactivity. And that means that, as you can see from this graph, from this slide, that deoxy sugars are really prone to undergo reactions via SN1 pathways. And the problem here with going through SN1 pathways is that for deoxy sugars, that leads to a whole realm of byproduct forming pathways, because if we by some way go through this SN1-like mechanism, we can imagine eliminations to occur either via E1 or E2 mechanisms. But alternatively, if our desired nucleophile starts reacting with this highly reactive intermediate, we actually get a very alpha selective reaction, which is exactly opposite what we want. This is governed by Fürst-Plattner rules. Additionally, if we then start forming this elimination byproduct, which is known as a glycal, the inherent problem here is that glycals are actually pretty good substrates for a named reaction, which is known as a Fourier rearrangement that yields 2,3 unsaturated sugars simply by an SN2 prime reaction, which is also not what we want. So we're really fighting not only a problem with reactivity, but also a problem with byproduct forming reaction pathways. So the question is, how have people addressed this in the literature? There's a whole bunch of papers out there on beta-2-deoxyglycosylation. I've selected two that I believe are the two most interesting and the two most important. So the first one I'm going to show you here is developed by Clay Bennett, also here in Boston at Tufts University. And you can ask yourself, how do you improve SN2 characteristics in a reaction? Well, one way of doing it is by simply using better nucleophiles. And that's exactly what the Bennett lab relied on, using potassium salts of their desired nucleophiles. And the way they simply go about this is taking a desired hemiacetal, reacting it with a sulfonic anhydride, in this case, toxic acid anhydride, to generate this tosylate, which is not isolated. It's not isolable. And then they just mix in a potassium salt of their nucleophile and get essentially beta-specific reaction, which is great. This has been used by other labs as well, and in the synthesis of really complex natural products like the lambamycin A hexasaccharide shown here, which is really incredible. The problem here, of course, being that it's under really harshly basic conditions. Another more recent development I want to show you is in a collaboration. So this was developed by Wei Li in collaboration with Bao Yu. And they, in this work, they rely on using this DPPA protective group, which they hypothesize acts as a directing group to direct the incoming nucleophile from the same phase as the protective group. So if this is pointing up, you're going to get beta-glycosylations. If it's pointing down, you're going to get alpha. And they have a really impressive scope of how to build oligosaccharides using this method. So I think this is really quite incredible. This was only published a few years ago. So we have an overall goal for our project here because we believe we fit in quite a special way in this field, because one method here relies on harshly basic conditions and another one relies on strong acid to promote the glycosylations. And we know our reactions react under essentially neutral conditions because it's just hydrogen bond donors. So we want to be able to have this neutral pH reaction going on at ambient or near ambient temperatures to facilitate beta 2,6-dioxyglycosylations. And we knew already that we would have to come up with some solution on how to lower the inherent reactivity of these electrophiles, and that we have a handle in choosing the phosphate leaving group to improve the binding of our catalysts. We know we want to be able to glycosylate both phenols and aliphatic alcohols because that really widens the scope of what we would be able to achieve. We want to have as high functional group tolerance as possible, at least gauge what the functional group compatibility is. So that means enolizable stereocenters, base-sensitive functional groups, natural products like peptides, and also basic amine-containing substrates. And we figured that amino acids might be very good model substrates. So in all of the optimization, you'll see me using tyrosine, and my collaborator Peyton mostly used threonine for his optimization. I won't be showing any of his results today, though. All right, let's get into it. So the first step of this project was to identify a suitable electrophile, because there are certain requirements for what kind of electrophile I could use in this project, because I needed for it to be stable and isolable. But in addition to that, it also has to be isolable as the pure alpha phosphate, because we're engaging in stereospecific glycosylations here. And in addition, it does not, or it's very important for us that it does not start decomposing up to temperatures around 40 degrees, because that's at least historically where we've seen our catalyst being most operative. And it also has to be reactive, of course, under HBD catalysis. So you could imagine synthesizing a bunch of different glycosyl phosphates that just happen to not be substrates for our hydrogen bond donor catalysts. So I'll summarize my search for an electrophile in this very simple slide. I cannot tell you how much pain is embedded in this slide. So I ended up finding this bis-trichloroacetyl protected neopentyl phosphate, as we refer to this leaving group, which is based off of neopentyl diol. But I've investigated a bunch of different protective groups and leaving groups, and in various combinations. So I think in total, I've probably synthesized about 25 different electrophiles. Some were okay, others spontaneously decomposed. This one was the best compromise of reactivity and stability. Then I have some initial results and optimization to show you here. So again, this is a very summarized optimization slide. So we've seen previously in our other glycosylation work that the handedness of the catalyst is really important, and in that was exemplified in this work as well. So this enantiomer of catalyst gave rise to a significantly higher yielding and slightly more selective reaction than its enantiomer, which really highlights the precise interactions between the catalyst and the electrophile, because there is definitely a recognition of the chirality of the substrate. I also tried using a simpler hydrogen bond donor like Schreiner's thiourea and found that that gave rise to an unselective but very sluggish reaction. I used twice the catalyst loading to account for that we actually have 2 hydrogen bond donors here and there's only one here. Or 2 thiourea, sorry, and there's only one here. I tried using TMS triflate as the promoter, which is a common way of activating phosphates that gave rise to an alpha selective pathway in line with what we would believe from the Fierst-Plattner rule. And in the absence of a catalyst, there's essentially no real background. I could only see about 3% of the alpha anomer in a group in the mass spectrum. A quick tour of selected optimization of the conditions of this reaction. We found that by lowering the concentration from 0.2 molar to 0.05 molar actually gave a slight bump in the beta selectivity in this glycosylation of tyrosine. However, and this was pretty interesting, this is the first time we've come across this effect, adding in basic alumina as a co-additive actually slightly improves the selectivity further, which was an effect we were pretty interested in understanding and that we've done some studies on since then. I unfortunately will not have time to show those today. I then tried running a reaction with 30 mole % catalyst to see if that would increase the conversion, and it certainly did. But also found that just using 10% catalyst and just extending the reaction time gave roughly the same yield. So I ended up deciding on these conditions as my optimal reaction conditions, and also showed that by adding in leaving group at the start of the reaction, that means phosphoric acid in this case, neopentyl phosphoric acid, did not have a really large impact on how this reaction works. And this experiment was really just to probe whether 4 angstrom molesits are an efficient sequester of phosphoric acid, which it turns out to be pretty efficient at. All right. So when working with these catalysts, we're able to especially tune the aryl groups. So on these aryl pyrrolidine scaffolds. So the slide I'll be showing here only optimizes or only focuses on the northern aryl pyrrolidine and keeps the southern aryl pyrrolidine. So we refer to these as northern and southern. This slide will keep the southern aryl pyrrolidine as this parafluorophenyl substituent. And we see by changing the electronic properties of the northern aryl pyrrolidine has some effect. So starting with this very electron-neutral parafluorophenyl to something more electron-rich or even more electron-poor actually results in mostly lower conversion, but also slightly lowered selectivity. However, by using extended pi systems, I started seeing some movement in the selectivity. So in particular, this 2-naphthol substituent gave a significant increase in the selectivity, whereas 2-anthracene and I believe it's 2-fen was slightly worse, gave lower conversion and worse selectivity, but 3-fen and 9-fen were pretty competent catalysts as well. And going for a very electron-rich aromatic group like this N-methyl indole resulted in a slightly worse reaction. And I have so in this case, 2-naphthol was the best substituent here. I have a few examples to show here of optimization of the southern aryl. We found that going from electron neutral to electron poor or an extended pi system made the reaction slightly worse. And I also tried some catalysts that have no substituent on the southern aryl pyrrolidine. So you can see there's neither a methyl or an aryl pyrrolidine here. And unfortunately, these 2 catalysts were also worse. The reason why I'm showing this is that this particular catalyst actually turned out to be the optimal catalyst in my collaborator Peyton's reaction. So in the end, I decided on this 2-naphthol substituted and 4-fluorophenyl substituted catalyst to be the optimal, and continued with looking into what the scope of this reaction was. The problem was, as I tried to scale up these reactions, I ran into some unanticipated problems. So after running columns to purify my desired glycosides, I started seeing unexpected amounts of this Fourier rearranged byproduct, which almost certainly goes via this elimination intermediate. And I also started seeing signs of trichloroacetyl groups being partially deprotected on silica. So you can imagine a TLC after a column essentially looking like I've been doing combinatorial chemistry. And I started testing a bunch of different methods on how to avoid this problem. I tried various different silica types to improve the purification, but in the end, the solution turned out to be direct trichloroacetyl deprotection after the glycosylation, and then just isolation of the deprotected sugars in the end. And very analogous to deprotection of an acetyl protected sugar, which typically happens under basic conditions in methanol, I was able to deprotect the TCA groups using very mild base, even as mild as bicarbonate. It has to be said that just pure methanol does nothing to this protective group, so the base definitely plays a part here. The only problem with this procedure is that I end up actually forming quite large amounts in some cases of this methyl glycoside byproduct, which happens after reaction with remaining glycosyl phosphate in solution. And this has its own problems, but I'll get to that. So in the end, optimal conditions here, I haven't shown you the optimization of the alkyl alcohols, but I'll show you now. So for phenols, we run these at slightly diluted conditions of 0.05 molar with this basic alumina additives. And for alkyl alcohols, it's just 0.2 molar, MOLSIFs, and this catalyst. And of course, after the glycosylation is done, I filter the crude, I add in base and methanol. I decided on sodium carbonate, and after an hour, I have quantitative conversion to the deprotected sugar that I then isolate. So let's take a look at the scope of this procedure. So I used 1.5 equips of glycosyl phosphate in all cases. And you can see that threonine and serine work glycosylated under the condition. However, threonine is a pretty sluggish substrate, and you'll see in general that compounds that have electron withdrawing substituents next to the desired glycosylation site is actually something that's really challenging for me to achieve. As you'll notice here, there's two yields shown. There's one without parentheses, that's the isolated yield, and in parentheses is the crude yield. The reason why I report both is that for some of these compounds on chromatographic purification, there was essentially perfect overlap with the undesired methyl glycoside byproduct, which made it really hard to get pure sample from some of these reactions. I managed to glycosylate some relevant glycosyl nucleophiles, so a 3OH and 4OH deoxynucleophile, managed to glycosylate a bunch of different interesting natural products and pharmaceuticals, namely chloromutylin, which again is a compound that has an electron withdrawing substituent right next to the alcohol, which gave an okay crude yield, but very low isolated yield. This compound was a perfect overlap with the methyl glycoside, and I could only get this out by preparative HPLC. And then ticagrelor is a compound that contains basic nitrogens, which is really cool that we're able to do that, because you could imagine this being challenging using, like, relying on strong acid as much of the state of the art does. I glycosylated a bunch of phenols, tyrosine, buc-serotonin, clofoctol, beta-estradiol gave a slight regioselectivity for the phenols, so a 2-to-1 mixture of phenolic and aliphatic alcohol glycosylated, and then tocopherol, androsterone, and this deoxycholic acid derivative were all excellent substrates in this reaction. And it has to be said that all the isolated yields and crude yields refer to this unprotected compound, just so we're all on board. And the same with the alpha beta selectivity is of crude spectrums of the unprotected sugar. All right, so this is the scope of the method. I'm hoping to be able to show this in a form of a journal very soon. Hopefully this will be available in some journal very, very soon. So before I end, I have some people I need to thank. So these are the people who currently work on glycosylation projects in the Jacobsen lab. So of course, I want to thank Peyton Beyer. So he actually started out as a student who I mentored, but I'm very proud to say that I really think that we are very equal in terms of, I see him as a very equal collaborator in the lab and a very equal contributor to this project. He's been working on 2-deoxyglycosylations. I worked on 2,6-dideoxy. He's standing right here. An incredible scientist. I also want to thank Agata Bikovtseva. She's standing right here. She's a grad student I also mentored in the Jacobsen lab, but worked on a completely different project that I will not be talking about. And then I also want to thank Zach. He joined very recently, so I haven't really mentored him for that long. And then also Elias was a postdoc who was involved in this project early on, but is now an assistant professor at the University of Southern California. I also want to thank Eric Jacobsen for giving me the opportunity to work in this lab. So here is me and Peyton, along with Eric at the more recent Christmas party at Eric's house. It's been an incredible opportunity to get to work in a lab. I really want to thank both past and present members of the lab. It's a pretty incredible atmosphere to work in a lab with such motivated and talented students. I can't wait to see where all of these folks end up in the future. Lastly, I also want to thank my former PI, Christian. He was really a key person in getting me to where I am today. He was always very open to discussing possible career opportunities and always trying to push me forward and just allow me to take chances, which has been absolutely incredible. And of course, I need to thank the Independent Research Fund for funding my stay here in Boston. And like I promised, I'll just give a quick teaser. I'll be coming back to the University of Copenhagen in April. I was lucky enough to receive a Carlsberg Reintegration Fellowship, which kind of sounds like I'm coming out of prison, which in some ways is also true, I guess. My research will focus on prebiotic chemistry and water microdroplets, so a very different field than what I've been working on previously, but I'm really excited to get started. And my initial focus will be on oligomerization of hydrogen cyanide formamide to form nucleobases or intermediates thereof, and try to probe a new hypothesis on how these might be formed under prebiotically relevant conditions. So just for those of you who might be interested, I'll be looking for motivated students on the bachelor's and master's level to join me in studying these, what I think are really fascinating chemical reactions. So if you want to learn a little more, you can always check it out on the Carlsberg Foundation's website, or you can just shoot me an email. I would also invite all of you to just, if you have any questions or want to know how I ended up in Boston, and you want to know how to avoid that or try that yourself, feel free to send me an email and just ask any questions. I'd be happy to help. I've already helped a bunch of students in their thinking about grant applications and stuff like that. I love doing these things, so please just reach out. So I think that's all for me. That was 27 minutes. I hope that's okay, Jens Christian. Thanks for your attention.